conclude our discussion of gene expression and transcription and translation, we have a few topics that will round out our perspective of the entire process. This includes talking about the way that introns are spliced out of the initially synthesized eukaryotic mRNAs, the transport of messenger RNA from the nucleus to the cytoplasm. We'll talk about how protein folding can be assisted by chaperones. We'll talk about how proteins are degraded, and we'll talk a little bit about the proteasome, which is the protein complex that degrades cellular protein. This is the anti-partner of the ribosome, if you will. And then we'll just talk a little bit about how antibacterial drugs target the ribosome as a way of trying to cause bacterial cell death. Here I'd like to briefly show just some of the data that was used for the discovery of introns. The experiment was to hybridize some messenger RNA to DNA to find out where the gene was. At this time, introns were unknown, and it was believed that the mRNA sequence was an exact copy of genomic DNA. If that were the case, you would expect it to hybridize smoothly as depicted here. But we know now that eukaryotic genes have introns in the DNA sequence, but that they are spliced out in the mRNA. Therefore, an mRNA will hybridize to multiple different exonic regions, causing part of the DNA to loop out. And these loop structures were observed over and over, and that was one way that the existence of introns was deduced. Just like the sequences that help proteins recognize where transcription should start and stop, there are sequences that help other proteins and RNA molecules recognize the exon-intron boundaries and where there should be a splicing event. Here there is an 8 base pair sequence at the 5' end of an intron and a 13 base pair sequence at the 3' end of the intron that indicates where splicing should occur. In the sequences here, in addition to A, C, G, and U, we see some other characters or letter. Y stands for pyrimidine, so either a U or a C is required at this point. R stands for purine, meaning it could be either an A or a G here. In the center here, you see some other required sequences that are involved. In particular, the A, which does some of the chemistry of the splicing reaction, in which the various RNA molecules can participate decisively. Splicing is performed by a protein and RNA complex called the spliceosome. It's a very large, complex machine. An example of the role that an RNA plays in doing some of the chemistry of the splicing reaction is that the base pair A that was in red on the previous slide attacks the upstream exon-intron boundary, forming this structure referred to as a lariat structure, which is then cleaved out. The two exons are joined, and now you have a spliced boundary region connecting two exons in your messenger RNA. This was one of the first examples of RNA having enzymatic activity, and the fact that we've now seen that RNA is a molecule that can both code for information, for instance in the viruses of genomes, and perform biochemistry like proteins. These dual capabilities has led some people to speculate on the possibility of RNA being the primordial molecules present at the origin of life. And that's as good as a hypothesis as we have right now. One of the differences between eukaryotic and prokaryotic messenger RNA that we've pointed out already is that the poly A tail at the 3' end of eukaryotic messenger RNAs. Once the RNA stop sequence for RNA polymerase has been passed, the RNA polymerase will dissociate from the DNA, dissociate the RNA from the complex, and an enzyme called the poly A polymerase, a polymerase which does not need a template by the way, will just start adding A to the end of a synthesized messenger RNA. This poly A run will be covered with the poly A binding protein, and it will be involved in the transport of the messenger RNA out of the nucleus. The fact that all eukaryotic mRNAs end in poly A has been of tremendous practical significance and useful because it allows scientists to take a poly T primer, which will hybridize to every messenger RNA in a cell and allow you to synthesize the complete set of them, making molecules that we refer to as cDNA or complementary DNA, and we'll see that in future lectures. Once synthesized and processed in the nucleus, messenger RNAs need to be transported out into the cytoplasm. In the nucleus, they're bound by the cap binding protein, Polya binding proteins, and complexes called the exon junction complex. Movement out of the nucleus is aided by a diffusion as it goes through the very large nuclear pore openings in the double membrane that separates the nucleus from the cytosol. Proteins that bind to the cap region of the mRNAs will help transport them to ribosomes at the sites where they are read and where they direct the synthesis of specific proteins. We saw earlier in the course that full-length protein sequences could be unfolded with urea and then refold on their own once the urea is removed. We argued that this demonstrated that the sequence contains all the information needed to direct folding, but we mentioned the caveat of kinetic trapping. Here is an example of that, kinetic trapping. As a protein is being synthesized, the amino end of a protein is exposed prior to completion of the entire polypeptide chain. So proteins can start to fold into domains before the entire sequence is even available. This shows you that kinetic factors potentially can play an important role in the final folded structure. If this portion of the protein starts to lock into a particular configuration, that once the full-length protein is present, it may not be able to unfold what's already been performed. And this is one of the reasons why we're not certain that that proteins fold to a global thermodynamic minimum, because these proteins can get kinetically trapped into a local minimum by the process where part of the protein starts to fold before the entire length of the protein is present. Sometimes proteins don't get folded correctly due to random events. Other proteins were never intended to fold entirely by themselves and require some help. A class of proteins that achieves this is called chaperones, and they can play large roles in folding many proteins and the correction of incorrectly folded proteins. When a protein is folded incorrectly, it will interact with a chaperone, which will then attempt to refold it, or it can also send it for degradation. The chaperone can try a few times to refold it, but after each attempt, there is a chance that the incorrectly folded protein will be marked for destruction. However, if a protein is folded correctly, then it will no longer interact with the chaperone and it can diffuse away to perform its cellular function. I think it's pretty amazing that the system can operate this way, and you might think for a few seconds about how the chaperone could possibly know if many different proteins were folded correctly or not. Please pause and we'll resume on the next slide after you think about it. Okay, here's one way how it knows. Proteins that are folded correctly proteins that are folded correctly have hydrophobic cores and hydrophilic surfaces. If a protein is incorrectly folded, there is a good chance that a hydrophobic patch that is meant for the interior of the protein will be exposed on the surface. Chaperones have hydrophobic binding regions, which can interact with any incorrectly folded protein that has a hydrophobic portion on its surface. Neither of these hydrophobic regions want to be exposed to solvent, so that they will be lower in energy if they are interacting with each other. This is a similar situation to what we saw with the globin protein in sickle cell anemia. Now, here, with the help of ATP binding and hydrolysis, the protein will be drawn into the interior. A cap will form, and now it's sort of isolated from other external influences, and it will attempt to refold it. It can then be released, and if it's correctly folded, it will diffuse away because it no longer has the exposed hydrophobic region to interact with the chaperone. But if it's incorrectly folded, it may by chance interact and go back again. This entire process of a protein entering the chaperone, refolding, and being released occurs in less than a second. And how the chaperone manages to unfold and let a protein refold, I'm not fully aware, but I suspect that it is not completely understood. The two cavities of a chaperone protein are essentially identical, though only one of the two cavities is active at any particular time. So here we see the protein complex called the proteasome. This is the complex that degrades nearly all proteins. It is also a very abundant protein in a cell. It can be 1 to 2% of the cell's protein mass. If you think about it, every protein that gets made by a ribosome can be degraded by the proteasome, and so that makes it have a very important role in the overall maintenance of protein level. In this sort of flowchart, it's mostly saying that protein aggregate is destined for the proteasome. But in actuality, any protein that the cell no longer needs that is marked for destruction gets processed by the proteasome. Ubiquitin is a small protein sequence that can get covalently attached to an existing protein. A single ubiquitin molecule, one of these red subunits, can have other functional roles, like for instance it can serve a role in signaling. There's a lot of different functions for a single ubiquitin. However, if there are multiple ubiquitin units on a protein, this protein is then marked for destruction. And that occurs because these ubiquitin units interact with the proteasome cap proteins. And again, it's just thermodynamics. If they encounter each other, they will bind, and if they bind, the protein will be threaded into the center of the barrel. Inside the barrel, there are 3 different types of protease activities inside the chamber that will digest the proteins into individual amino acids, which then can be reused by the cell for additional protein synthesis. It's still a very active area of research in terms of figuring out how the cell knows which proteins should be tagged for destruction if they're properly folded and simply no longer needed for utility. But the basis of most of this is controlled by 3 types of proteins that form the ubiquitin ligase machinery. The subunit types are called E1, E2, and E3. Human genomes have a few E1 units, maybe a dozen or so E2 units, and maybe 6 or 7 dozen E3 units. And so combinatorially, you can have potentially many different E1, E2, E3 complexes. And each of them has the potential of targeting different proteins. We are, however, a long way from understanding which complexes target what proteins and for what reason. In any case, it's the E2 unit that will transfer ubiquitin to the protein it's going to mark for destruction. Additional ubiquitin subunits will be added to the chain, and now this protein will soon be degraded. Here we see the bacterial ribosome and highlight many of the antibiotics that target the ribosome in an effort to kill bacteria. Bacterial ribosomes and eukaryotic ribosomes are fairly different, so that when you take tetracycline or erythromycin, they will leave your eukaryotic ribosomes alone, but they will bind to the bacterial ribosomes. You may know that when a drug is given to bacteria, bacteria will try to mutate so that the drug will not bind to it and so that it can escape cell death. But the ribosome is such a complex machinery which has to be so careful in recognizing with great fidelity each of the 61 different tRNA molecules that it doesn't have a lot of room to maneuver in terms of mutations to avoid being bound by the drugs and still function properly in terms of making proteins. But as you know, there's resistance to nearly every antibiotic that we have, and so the bacteria have found ways to escape these drugs. But it's still an attractive target because of the many constraints on the design of this complex. I include this slide as a review for all the things we've talked about: transcription, processing of mRNAs, translation, protein folding, protein degradation. This slide is a good review for you because it captures a lot of the information as you prepare for the first midterm. Okay, thank you for listening.